Okay, so here's the material for lipids. There's been three previous videos put out. One on polarity, carboxylic acids, esters, and molecular interactions. And now we're going to see how those apply to lipids, our second biological macromolecule. And so let's get with it, and hopefully you'll see how that integrates with those three videos. Okay, this video is probably going to be about an hour. Oh, it's almost 2 hours long. Okay, and so in the announcements I showed how you can break it up, and don't try to take it all in one chunk. It's just too much. And download the no voice version. Good idea if you can't get a hard copy, at least download it on your computer. But to get yourself a copy of it, and then what you can do is you can take notes on the no voice version as you go along and listen to it. And I would suggest break it up about 30-minute segments. So if you do that, there's 4 segments. And the test is coming up in a week, in about a week and a half. So okay, so lipids are nonpolar, and that means we went over that. We talked about nonpolar when we talked about polarity, and nonpolar molecules are not normally very soluble. That means they don't dissolve in water, and we'll explain why that happens. We said that what happens is when you drop like oil, it's nonpolar. When you put it in water, the water will try to get around the oil molecules, and doing that will push the oil molecules together, and that causes them to have better interactions under dispersion forces, and so they stick. And so it looks like oil and water don't mix, and what we say is oil is hydrophobic, but not really. It's just that water enjoys being with itself better. So lipids are nonpolar. Don't dissolve normally in water. So now we talked about how you can tell if you have a nonpolar molecule, and there's 2 ways. One way is to look at this if you're given the bonding structure, and if you look at the center atom. So if you have something like this, okay, these are just. So you have a center atom B, and if the center atom has the same things attached to it, it's nonpolar. But in this case, it has different things, so this would be a polar molecule. But if it had the same things, like a B and 2 Ys, then you could identify that it's not polar. It doesn't tell us that it is why it's nonpolar, but it is. It identifies that it is. The other way is if you have a molecule and you look at the chemical formula, maybe like CH. The molecules we deal with have a lot of carbons, oxygens, and hydrogens. So I said if you had something like this, C3H1425 or 2, I said if the ratio was less than 3:1 carbon to oxygen, then it was polar. If it's greater than 3:1, then it's nonpolar. So a molecule that has a lot of carbons in it, you can probably guess compared to oxygen that it's nonpolar. And that's what you're going to be noticing here is that these molecules have lots of carbons compared to oxygens. And there's 2 types, fatty acids, and then there's steroids. They look completely different, and what makes them similar is they're nonpolar, which means they have lots of carbons in them. Okay, so let's move on. Okay, so here's the general flow of what we're going to cover. So we're covering lipids, which are nonpolar macromolecules, and that means they have way to define that they have a lot of carbons. Okay, there's 2 types: fatty acids and steroids. Okay, we're going to spend our time talking about fatty acids first, and then finish off with steroids. Okay, so let's look at compare these 2 guys right here and see how they're similar but alike. Okay, so every point on the zigzag line is a carbon. Okay, so this guy here has 1, 2, 3, 4, 5, 6, 7, 8, 10, 11, 12, 13, 14, 15, 16, 17, 18 carbons and only 2 oxygens. So the ratio is way bigger than 3 to 1, and you can see that therefore it's nonpolar. Okay, and steroids, every one of these points is a carbon, and sometimes there's oxygen coming off, but mostly steroids are carbons. Okay, so that's what's common between them. They're non-soluble, and the basic reasons because they have a lot of carbons. Differences, you can look at the structures. All right, so let's look at fatty acids. First, we're gonna do is look at study fatty acids a little bit. Okay, in the first video, I talked about carboxylic acids. We talked about that's that COOH. C. Okay, has a carbonyl. Okay, and it has a hydroxyl. Okay, so carbonyl, hydroxyl. So carboxyl has a combination of the words. We'll talk about that in a second. Okay, and it has a long, and so it's a carboxylic acid with a long carbon chain. All right. So we're going to look at different kinds of fatty acids, and then we're going to look how they play. Okay, so we find fatty acids in waxes, we find them in triglycerides, we find them in soap, and we find them in phospholipids. And we'll talk about that. And then when we get done with those guys, we will talk about steroids. Okay, and we'll talk about cholesterol. Not so much bile salts, but bile salts are a result of cholesterol. And then steroid hormones. Okay, so that's the general flow of our material. Okay, so fatty acids. Okay, again, we find them in triglycerides. They're all by themselves, they're fatty acids. Sometimes you might in our blood. Sometimes they're found all by themselves as they're called free-floating fatty acids. But they're also found in groups: triglycerides, waxes, soaps, and phospholipids. And there's steroids, which I just talked about. Okay, so let's get into fatty acids. So fatty acids are long-chain carbons, carboxylic acids. And they have between 12 and 18 carbons. And usually they're even numbers. Okay. And there's 2 types, saturated and unsaturated. Now, in the very first material we talked about alkanes. And if you go back and look, okay, you will see that alkanes were called saturated hydrocarbons. Okay, a saturated means is that all the carbon bonds are single. And all the other bonds are hydrogens to carbons. Okay. So what we say, it's saturated with single bonds. Okay, for saturated fatty acids, the long chain carbon will all be single carbons. Okay, I mean single carbon bonds. Okay. Unsaturated, when we talk about the alkenes, okay, it didn't have all single bonded carbon bonds. They had at least one double bond in them. So they're not saturated with single bonds. All right, they have they're not all filled up with single bonds. They have at least one double bond. And the same thing that's going to be found with unsaturated fatty acids. Unsaturated fatty acids are going to have the long chains will have at least one double bond in the carbon chain. Okay. Now, one of the things we'll notice, and I'll show this in a second, is saturated fats have higher melting points than unsaturated fats, and we'll explain why that is. And saturated fats tend to be solids at room temperatures, which we'll talk about in a second. All right, so let's look at saturated fats in different formulas. We have 4 saturated fats. lauric acid, which is the main. So if you had a coconut and you open it up and look at the white material, most of that is lauric acid. And then we have myristic acid, which is found in nutmeg, and so forth. All right. Okay. Now, if you look at each one of these, is that we increase the number of carbons by 2. Okay, that means this chain here increases by 2. This is the carboxylic end, okay, and this is the long chain. Right, so 12, 14, 16, 18. Now, let's look at, you can see that they physically look different. This guy's longer than this guy down here. And if we also look at their melting point, we can see that the melting point is directly related to the size of the length of the chain. Okay, so that's what we want to explain, is why that happens. Okay, so this is in Celsius. Let me just give you a little idea about what temperatures. 43 Celsius is about 105, 110 degrees. Fahrenheit 54 is about 140 degrees. Fahrenheit 60 is about 155, and 69 is about 107 degrees Fahrenheit. So it takes quite about a heat to melt animal fat compared to coconut, which is just slightly above room body temperature. Okay, first of all, let's make sure you understood what melting means. Melting point. Okay, melting is when you have a solid, and what a solid is. Okay, so a lot of people get this mixed up. A solid's when a bunch of atoms or molecules interact with each other and form very tight interactions, and that tight interactions cause them to be a solid. Okay, now when you add energy and turn it to a liquid, those interactions break apart. Okay, so it takes energy or heat to break apart the interaction between the atoms or molecules. It doesn't break the molecules, it breaks them apart. So if, for instance, you have ice and each one of these dots represents a water molecule, okay, and in ice they're all connected, and then when you add heat to melt them, these molecules will separate from each other to form a liquid. Okay, so when you melt a solid, you're breaking interactions between molecules, or atoms, in this case molecules. So if you look down here, it takes more energy. Temperature can be related to energy or heat. The more energy you need to break these molecules apart. So what is it about this guy that requires to break apart the interactions of many of these guys? Okay, the way that I look at it is I kind of think about spaghetti. So let's say you have a bowl of spaghetti and you've cooked it up, okay, and you have maybe 20 strands of noodles, and they've all been cooked up, and they're all in a bowl. And then you have also cook up 20 noodles of macaroni. Okay, and what I ask you to do is I'm going to ask you I'm going to give you one bowl, give you the 20 spaghettis, and find out how long it's going to take you to separate each one of these spaghetti strands from each other. Okay, and then I'm going to do the same with the macaroni. Okay, so which one is it going to take longer to separate, or which one's going to take more energy, in a sense? Well, hopefully you'll see that it takes more energy to separate the spaghetti. And the reason why is because they're longer and they stick and they tangle. Okay, they're able to wrap around each other. The smaller ones can stick, but they can't wrap around each other. So that's the same thing that kind of happens with these. The longer the carbon chain, it more acts more like spaghetti. And the smaller, more acts like macaroni. And so it takes more energy or more temperature to separate these guys and these guys. And that's why the long chains are harder, needs more energy to separate them than the small ones. Okay, and so I go over that, okay? I sometimes call it the tangling effect. All right, now let's look at unsaturated fats. So unsaturated fats have at least one double bond, and let's take a look at this guy right here. Here's palmitoleic acid. It's found in butter, okay? And you can see it has a double bond right there. One double bond. You can draw it either way, this guy up here or this way. Oleic acid has one double bond as well. Okay, and notice you get the similar time effect is that this guy has 16 carbons, this one is 18. This oleic acid requires more temperature to separate it. Okay, that's because it's longer. Okay, now, so you can also have, so these guys are called monounsaturated because they only have one double bond. But you can also have a polyunsaturated, and that's where they have at least two double bonds. Like linoleic acid has two double bonds. Linolenic acid has 1, 2, 3 double bonds, and arachidonic acid has 1, 2, 3, 4 double bonds. Okay. All right, and these are polyunsaturated. Poly means many, so it's many double bonds. Okay, now one of the key things to notice here, they all have the same, well, these two guys have the same, these three right here all have the same number of carbons, but if you notice, is that the more double bonds you have, the less temperature you need to melt it. Okay, and so we're going to look at that next. All right, so let's take a look at how we have, so there are 4 molecules in the previous 2 slides that had 8 carbons. Stearic acid, okay, that had and oleic acid, linoleic acid, and linolenic acid all have 18 carbons, but if we look at the boiling point, they all have different boiling points, okay? So it takes more energy to separate or melt the stearic acid than this guy down here. Okay, even though they're the same lengths. All right. So if you notice, there seems to be a correlation between the number of carbons, double bonds, and lowering the energy required to melt it. And okay, so what is the reason for that? What does the double bonds do that somehow decrease the tangling effect? Well, the double bonds, what they do is they stiffen up the chain. So it's like having kind of like uncooked spaghetti. So if you uncook spaghetti, so if I give you 20 pieces of spaghetti, I only half cook it, it won't stick together better, okay? And it's either easier to pull it apart. And that's sort of what happens here is these double bonds act like they stiffen the chain, reducing the tangling effect. So more double bonds reduce the melting point because they reduce the tangling effect because they're more stiff. Okay, so here you go. And so it's explaining it. Okay, now what we're going to do is we're going to talk about our fatty acids, and then we're going to put them in waxes, and we're going to put them in triglycerides, put them in soaps, and finally phospholipids. So the first one are waxes. Okay, and waxes are esters. Okay, so we talked about esters in the very first video of this material. Remember, ester is you have a carboxyl group. Okay, but instead of having an H over here, you have a carbon chain over here, and you have another carbon chain over here. And so, and this part comes from an alcohol, and this part will come for carboxylic acid. You won't have to know that. You just have to know that a wax and an ester. So it has a carboxyl group, where it has one carbon chain and one carbon chain over here. So these guys have they have lots of carbons. They have lots of carbons on this side, lots of carbons on this side as well. That means they can have up to 40 carbons. And if you can have that many carbons compared to the number of oxygens, these guys can be really hydrophobic and nonpolar. Okay, now where these are found in nature is you find them in plants, and they'll be found on like succulent plants, and for instance like cactus, and the cactus will have this waxy material that covers out the outside of the leaf. And the reason why that is that when the cactus leaves will have water on them, and if they're in the middle of the desert, that water, if it didn't have that waxy covering, that water would evaporate into the air. And so what happens is that the water will try to escape out of the leaves, but because water and waxes don't mix, the water is pushed back inside the leaves, and it stays inside the leaves. All right, animals, we have like earwax, but the kind of earwax type things on our skin, our fur and feathers, okay, and maybe not. We don't technically, you might know them as waxes, but they're like oil waxes. And the reason why we have them is basically to keep warm, because in the winter, as you move around, you will start to sweat, and as you start to sweat, that sweat is holding heat. Okay, what you want to do is keep that heat in. So what happens is that as the water tries to escape with heat out of the skin, the wax will repel the water and therefore keep the heat in. All right, here's some examples of some waxes. All right, notice here's our carboxyl group, and then we have 15 carbons over here, and then over here we have 30. So overall there's 46 carbons. So this is very hydrophobic, okay? And this is found beeswax. This is found beeswax, and they use it for these right here. And you have this guy, carnauba wax, used for furniture, cars, shoes, etc. This one has 25 carbons and 30 carbons, so this is like 55, 56 carbons. So very hydrophobic and holds back water. So when you if you put this on your car, and then you spray the water in your car, the water is just going to beat up. It can't get through the wax in your car, because this non-polar parts will, in a sense, kind of repel the water. Okay. All right. So there's waxes. Now what we're going to do is we're going to take our fatty acids, and then we're going to put them in a molecule called a triglyceride. Okay, that's this guy right here. A triglyceride is a glycerol, which we learned about in the last material. Okay, we'll show a structure of that. And it has 3 fatty acids attached to it through ester bonds. Okay. The triglycerides can be broken up into 2 types, fats and oils. Alright, so it's called triglycerides because it has 3 fatty acids attached to glycerol. And we'll go into more specifics. Okay, so let's talk about the formation of a triglyceride. So first thing you need to have is glycerol. And we talked about that in carbohydrates, because this is the alcohol. It has OHs attached to it. And then what we're going to do is we're going to do esterification. And what we're going to do is we're going to add 3 carboxylic acids. We covered that in the first video of this material. And so what we're going to do is we're going to knock off the OH of the glycerol, and we're going to knock the H off of each one of the fatty acids, just like we did when we talked about forming esters. Those 3 guys are going to form 3 waters. Okay, and then this will snap together with this. This will snap together with this, and the CH2 will snap together with this, and then you end up with this guy right here. And then you have 3 ester bonds. Okay. All right. This is a triglyceride. A triglyceride is a glycerol with 3 fatty acids, and a water is produced. Okay. So we're going to study this a little bit longer because there are different kinds of triglycerides. Okay. So there's different types of different kinds of triglycerides. And what we do is we look at the fatty acids. So the fatty acids on the triglyceride they can be different. Okay, you can see that this glycerol has one with a 16, and this one has a double bond, and so forth. Okay, well, this triglyceride has all the same fatty acid attached to it. Now, the type of fatty acids attached to the glycerol is going to determine the type of triglyceride. Okay, so the first type of triglyceride is called a fat. And by definition, by our definition, a fat is a triglyceride which is a solid at room temperature, which is about which we say is 70 to 75 degrees Fahrenheit or 20 to 25 Celsius. Okay, these would include like the fat on your body, animal lard, butter fat, Now, these will have, because they tend to be solids at room temperature, that means so when you look at the 3 fatty acids, 1, 2, 3, most of the time, at least 2 of these will be saturated, fatty acids, all right? So maybe all 3 of them. Remember, saturated fats have higher melting points, and they're more likely to be solids. So since you have more fatty acids are more likely to be solids on this thing, this whole thing is more likely to be a solid. So if you have at least typically 2 or more, 2 to 3 saturated fatty acids will be found on fats. Oils, on the other hand, like vegetable oils, not like motor oil, we're talking about biological oils, are defined as triglycerides that are liquids at room temperature. These are usually found in plant sources, vegetable oils, And what makes them vegetables is the triglycerides, the fatty acids, tend to be unsaturated, Again, so somewhere between 2 to 3, 2 of the 3 or all 3 of these will be unsaturated, which means they have at least 1 double bond. So most fats they don't have to be all, like I said, they could be 2 of the fats are saturated or 2 unsaturated. All right, so here's a profile of different fats and oils, triglycerides. And let's look at the overall trend here. So at the top here, have the highest melting point. That means they tend to be solids at room temperature. So these three here are probably are found at solids at room temperature. Okay, these down here are found as liquids at room temperature. All right. Now, if you look, so here's a little key down here. Yellow, poly, unsaturated. That means a lot of double bonds. Mono, one double bond, and blue, no double bonds. If you notice, these three up here, have a lot of blue. That means they have a lot of saturated fatty acids in them, which makes sense. Remember, saturated fats tend to have higher melting points. And if you look at these guys down here, the blue, there's a lot less blue and a lot more yellow and red. Okay, so they will have lower melting points. Okay, now one of the things that's interesting about this is what's this important in terms of our body? Okay, okay. So we're gonna look at triglycerides in our body, and I'm not gonna do a giant physiological study of this. You may learn this in your A&P class or biology class. But I just want to give you a background how important these are and what their function is. We just went over their structure. Let's talk about the function. One of the things so one of the key things about these kinds of molecules is that these molecules, fatty acids, are really high packed in energy. Okay, and so I think it is that for every 1 gram of carbohydrate or protein, okay, 4 calories of energy will be used to break that down. So you can get 4 calories out of 1 gram of carbohydrates. For these guys, for 1 gram of fats, you get almost twice that. You get 9 calories. So fats are way more energy efficient than carbohydrates, proteins. Okay, so so one of the function is what triglycerides do is they move fatty acids to the cells. So remember, a triglyceride has 3 fatty acids on it, and what happens, we'll get to this in a second, is that triglyceride carries these 3 guys to the cells, and they get dumped off in a cell, and they'll be used for energy. Another place is fatty acids. If cells can't use them for energy, they store them as adipose tissue, and so that's like the fat on your body. And here's that as well. Right. So let's look at sort of the progression here. So let's say you eat most of the fats you get from your food are in triglycerides. Okay, I mean the fatty acids. Okay, so when you eat some food and it has fat in it, as we call it, it has triglycerides in it, either fat or oils. Okay, they go into your gut, and when they get into your gut, they go from your stomach to your intestines, and then finally into your blood. Now, the reason why they go in your blood is so they can move them to your cells. Okay. Now, if your body says it needs energy, okay, the cells will take in the triglycerides, break the fatty acids off of the glycerol, which we'll talk about in a minute, and the fatty acids will be released. And the fatty acids will go through a similar pathway as cellular respiration to make ATPs. Now, if for some reason your body doesn't need that energy, okay, maybe it's using carbohydrates or it has a very low energy state, it will send it called the adipose tissue, and the adipose tissue will store the triglycerides. Okay. And then, if for some reason you run out of food, you're not eating, okay, your body says, I need energy, and what it'll do is it'll release the triglycerides from your adipose tissue to your cells, and then the triglycerides will be broken into their fatty acids and released as energy. All will be broken up to make ATP, which is you can use for energy. Okay, so when the triglycerides get to your cells and need to be broken off, so I need to break these guys off, okay, so they can be used to be broken down to make ATPs. So what happens is that when they enter the cell, water is added to this guy, and it breaks apart the glycerol and the 3 fatty acids. And then this is called hydrolysis. Hydro means water, lysis means break, so break with water. So hydrolysis will break an ester bond. And now you have the 3 fatty acids, and the 3 fatty acids can be used to make energy. Now, if you notice, this is the reverse reaction of esterification. So if you take your 3 fatty acids plus your glycerol, you'll make your triglyceride and water. So this is the opposite of triglyceride formation. Okay, now, so we're going to move on to the next thing. We're going to talk about a couple reactions. That has to do with fatty acids and triglycerides. One's called hydrogenation, that we've talked about already, and one is called saponification. Okay, so let's look at this reaction called hydrogenation, and we've seen this already. Okay, this was on the first test. It's when we took an alkene. All right, and we added H2 to it. Okay, so hydrogenation, I mean, it means adding hydrogen. And what happens is the double bond disappears. Okay, so what you do is you take unsaturated and you end up with a saturated. Okay, so in the first test we talked about taking unsaturated hydrocarbon, hydrogenate it to make a saturated hydrocarbon. Well, you can do the same thing with the fatty acids because there's saturated and unsaturated fatty acids. So you can take a unsaturated fatty acid, one with at least one double bond. You can add hydrogen to it, and you can make it a saturated fatty acids. And this is an industrial process. This doesn't happen in our bodies as far as I know. So let's talk. Okay, so here's the reaction. And so here's for example, if you had triglyceride, you can take these double bonds here, and you can add hydrogen, and then you can get rid of the single bonds. So all the single bonds are gone. So you have unsaturated fat going to saturated fat. Now, if you look at this, unsaturated fats are oils. Okay, make oils usually, especially if there's 2 or 3 of them. That means they're liquids at room temperature. Okay, after we get done with this process, what we end up with is a fat, which means a solid. So what you can do is you can take a liquid oil and turn it to a solid fat. Now, what good is that? Okay, the reason why we do that industrialize is that we can take vegetable oil, which is a liquid, maybe like olive oil or whatever, canola oil, and we can hydrogenate it. And then what we end up with is solid okay, fats, or maybe like shortening, which is a replacement for things like lard and so forth. So this is how, margarine. This is how they make margarine. This is how they make Crisco and other shortening. Okay. All right, and there's some other things as well that happen with that. So when it undergoes this process, is that you can actually mess up the chain a bit. But there you go. Let you read about it. Okay, the next thing to talk about is making soap. And we're going to talk a little bit about the chemical reaction, how to make soap. And then we're going to talk about how soap works. Okay, so soaps are fatty acids or triglycerides that have fatty acids. And they have an electrical charge on them. Okay? So they contain long carbon chains, which are nonpolar parts, that don't dissolve in water, but they have an extra feature. They also have a part to charge that's dissolved in water. So if you had your typical fatty acid, your typical triglyceride, each one of these lines here are really hydrophobic, and when you put this in water, it wouldn't mix with water. But when we make soap, what we do is we take this guy, and what we do is we do a chemical reaction where we charge up the end of this, okay? And so what you end up with is a nonpolar tail, okay, which doesn't mix with water at all. And then you have a polar head, which likes water. And so you have sort of this dichotomy of parts. You have a part that likes water and a part that doesn't like water. And we'll see how that helps explain how soap works. And so this is kind of what you would look at. So you'll see that this is a nonpolar part, and we could just say it does interact with water, hydrophobic, nonpolar. And then it has this negative part that likes water. All right, so let's see how this works, how soap works. Okay, so when you take that long chain with the charge on and nonpolar, okay, when you put these soap molecules in the water, what happens, just like in oil, the water will push these tails together because they're nonpolar. And so what you get is all the nonpolar parts being pushed together. Again, when they get pushed together, they will increase their London dispersion forces and like each other better. Okay, so this pushing together by water causes these tails to form London dispersion forces. Okay, now there's another type of interaction. So there's two types of interaction. So there's the tails interacting with each other, and then there's other interaction with the heads, or forming dipoles, which we learned about. With water. Okay, so they like water. So this whole thing will actually dissolve in water because it's outside likes water. Okay, but it has a cavity or inside that's sort of hidden from the water. That's held together by London dispersion forces. This structure is called a micelle. Okay, so here's a little cartoon of it right here. Okay, so all these tails are interacting through London's dispersion forces, and the heads are interacting with the water through dipole interactions. So how does soap work? Okay. Okay, so how does soap work? All right, so when you have your soap molecule, okay, you put them in water. Okay, so all right. So when you wash your hands, okay, what happens is the oils of your hands are kind of just explain it. I'll just say it like this: the oils from your hands, which hold all the dirt and everything, okay, get trapped inside this center, inside the non-polar parts. So oils of your hand are non-polar, and the non-polar parts get trapped inside the non-polar of the micelle. And so they sort of get hidden inside there. Now, I'm not sure if this is the greatest analogy for you guys or not, because I don't know if you know what Trojan horse is. But, like if you ever read the Iliad or whatever, okay, ancient story about Greece, was when I think it was Greece, and they go into Troy, and they want to give a gift to the enemy. And the gift is a horse, a wooden horse, this giant horse. And inside the giant horse is our soldiers. But no one knows them. Okay, so they accept the horse, and they don't realize it's what's inside it, the soldiers. And that's kind of what this is here. Is that the outside world doesn't know what's inside here. Okay. And so the outside, these polar parts, they just see the water. Water just sees the polar heads. All right, so here's a three-dimensional picture of it, kind of. So inside the micelle is where you have your oils from your body, and they're trapped in there. And when they're trapped, those oils have got germs and dirt and everything. Okay. But the outside world, the water, is not going to see that. Okay, it's just going to see this, and so all of this is hidden. All right, so let's talk about how soap works. Okay, and let's go through the process. So when you wash your hands, the first thing you should do is you should take some water, and you should dampen your hands with water. Okay, it doesn't have to be a whole lot. Next thing you do is you squirt the soap in your hands. Okay. Now, why did we put the water on first and then the soap? So when you put the water on, and then when you add the soap, what's going to happen is it's going to start to form these micelles. Okay. All right. So this micelles start to form, and then what you do is you start to agitate the soap and water in your hand by moving your hand back and forth. And what's that going to start doing is it's going to start uplifting the oils from your hands. Okay. And the oils will get trapped inside the micelles. Okay, because that's the oil part, right? Okay. Next thing you do is now you have all these micelles that look polar to the water. So the water thinks these guys are just like them. Okay, polar likes polar. Right. So next thing you do is you take some water and you rinse. And when you rinse, what that water is going to do is that water is going to grab a hold of these guys and carry them away, because these guys like water, and they're going to go with water, and they go off your hands and into the sink. Okay, and therefore you should have clean hands. Okay, next category, and the final one of dealing with fatty acids, are phospholipids. So we covered waxes. First covered fatty acids, two types, saturated and unsaturated. Then we applied it to waxes, and then we applied it to triglycerides, and then soaps, and now phospholipids. Okay, a phospholipid is a lot like a triglyceride. Okay, so a triglyceride had 4 parts. It had a glycerol and 3 fatty acids. Okay, a phospholipid is going to replace one of these fatty acids with a phosphate that has a charge on it. And the way that we draw that is like this. Okay, so here's our glycerol. This is a fatty acid. This is a fatty acid, and this is a phosphate. This phosphate will have a charge on it, okay? Say a negative charge. So it's kind of like soap. It has a polar head and two nonpolar tails. Right. And so what we can do is just like soap. If we take many of these guys together, what's going to happen is similar to soap. Okay, the nonpolar part, tails, will aggregate and form London dispersion pores, so we'll get more in depth in that in a second. And the heads will interact with water. Okay, and we'll see this structure. Okay, and we can kind of see it already. Is that it has two layers. It has a layer here and a layer here. And we'll see that how that applies in a minute. Okay, so here's again, this is the idea. So here's your triglyceride or triglycerol. We'll call it triglyceride. And again, replace one of these guys. This guy with phosphate. And now this guy has a charge on it. These guys are nonpolar, hydrophobic, hydrophilic. Again, this is a more comprehensive picture of it. Okay. All right. Where are phospholipids found? They're the most, they're found in your cell membranes. Okay. And anywhere where there's a cell membrane, okay, like your regular cells, and also in your myelin sheaths that are like found in your spinal cord and so forth. They're there to protect the nerve cells, also to help nerves work better, okay? And we'll see how this all forms in a second. Okay, so when you take a phospholipid and put it in water, like I just drew before, you're going to get this type of thing happening. And water over here, and water over here, and this is actually an important thing. Where the nonpolar parts, the tails will interact. Now, they interact two different ways. They'll have, the ends will interact, but the sides will interact. So it's a stronger interaction than soap. Because soap just has end-to-end interactions. Okay, these guys will interact side to side. So London dispersion force, it has two types. It's tail to tail or end to end, and side to side. Okay, and all of that increases the London dispersion forces. So it really helps hold this thing together. Okay, all right, these two layers together. The other thing is the heads will interact with the water and form dipole-dipole interactions. So phospholipids, they form a bilayer when you put them in water, where they're held together by London dispersion forces, LDFs, between the tails, both end to end or tail to tail and side to side, and polar heads with water dipole-dipole. Okay, and here's another diagram of that. Okay, so the nonpolar parts will interact end to end and side to side, and the polar heads will interact with the water dipole-dipole. Now, if you noticed, okay, if you keep on doing this forever this way and forever this way, you basically have a wall, all right? This bilayer can be a water that separates this side of the water from this side of the water. And that's essentially what happens in a cell. A cell is going to form a phospholipid bilayer where one side is going to be outside of the water, outside of the cell, and then the other part will be the inside of the cell. So this guy, these phospholipid bilayers, is a way of forming a wall between the in and out of the cell, which is a really good thing because you don't want the inside of the cell to be messed up by the outside of the cell. It's got to do its own jobs. But we're going to see in a second is that there has to be some communication from in and out. But let's get to that. We'll get to that in a minute. So cell membranes are composed of 2 layers of phospholipids called phospholipid bilayers. And here's a good diagram of it. Okay, so you can see all these. So you have your heads and your tails. And heads and your tails. Okay, over here is the outside. Over here is the inside. Okay, so you have your inside water and you have your outside water. And technically, if you had nothing else, you could totally separate the outside water from the inside water. So no water could get in from the outside, and no water from the outside in could get out to the outside. Because any water trying to penetrate would bump into these, can't get through the nonpolar. Okay, but we know that we need to have a flux of in and out of the cell, but it's got to be controlled, all right? And so what we got to do is we got to add things to the outside of the cell inside this membrane that can help that flow. And this overall structure is called the fluid mosaic model. Okay, so there's going to be some key components inside the membrane besides phospholipids. Okay, and we'll go over each one in a little bit of detail, but not a lot. There'll be carbohydrates and cholesterol, and we'll go over each one of those. There'll be cell receptors and proteins. So let's take a look at the pictures and describe what we got. So if you look at this, is a real simplified version of it. All right. Now, inside this, besides phospholipids, there will be cholesterol. And cholesterol is there to help stiffen up the cell membrane, so it doesn't flop around very much. It's like putting a piece of plywood in there. It helps stiffen up the structure. Okay, so cholesterol is there to help stiffen up the structure. Has carbohydrate side chains, which will act like signals. So maybe like they'll send out information, or just to structure them will tell other cells what they are. For instance, different the different blood types A, B, and O. The reason why they're different is that each one of the different blood types will have different kinds of carbohydrate side chains on them. So A will have one kind of carbohydrate on it, B will have another, and O will have another. Okay, so these these side chains help tell the outside world, what kind of cell this is. There's also called these guys here are called integral membrane proteins, and they're for different reasons, maybe for cell receptors, where certain molecules can attach and relay information. Okay, here and here's one of those. Okay, here, all right. Another type is, and so here is the hydrophobic part, okay, which is the tails. Okay, and these guys here are going to be channels. And these channels are, and here too, is where certain molecules can move in and out. And we're going to talk about that mobility in the next slide. All right, for things to move in and out of the cell, which is necessary, okay, but you have to be very specific. Okay, you just can't let everything in and out of the cell. First of all, this membrane will not let things in and out very well, okay, because of its structure. And the nonpolar will, if you try to pass a polar molecule through it, it won't. It just won't go through it because of the nonpolar tails. Okay, but some molecules can pass through kind of easily, like oxygen and carbon dioxide. Okay, and that's important, that they can just move in and out without any problem. Okay, and that's because we constantly need oxygen to go into the cell, and we need the carbon dioxide to leave the cell. So we don't want to, we just want them to, they should have free access in and out of the cell. And the reason why they can do that is because they're nonpolar and they're small. They can squeeze in and out. But they're bigger proteins like glucose, fats, and other things that won't fit and need some help. And so let's look at that. Okay, so there's different ways that molecules and atoms can get through the cell membrane. One is called diffusion. That's like oxygen and carbon dioxide. They can just fit through, okay, and partly because they're small and hydrophobic, so they can go in and out without much deterrence. Okay, bigger molecules. There are two different ways they can go through. One is they can just go through this tunnel without much hindrance because the tunnel is made specifically for them to get through, maybe like a glucose tunnel. And so when glucose comes in, okay, the glucose, it will find its own specific tunnel and it will go through into the cell. And you need to have glucose. For instance, insulin might come open up this tunnel and let the glucose in. Right. Now, there's some guys that they need to pay a toll to get in and out, all right? And so we call that active transport, all right? And active transport's when an atom or molecule has to pay a toll to move through, and that toll is ATP. So the currency for in chemistry is ATP. So you have to spend some ATP to move in and out. Okay. All right. So we're done with the fatty acid section. Now we go to the steroids in the last section. Okay, so let's look at the basic structure of all steroids. This basic structure is called a steroid nucleus. They'll all have this in common. They'll have this structure. They'll have 1, 2, 3 cyclohexanes and 1 cyclopentane. Each point represents a carbon. All right, and what we're going to do is we're going to end up putting oxygens off the side and so forth. But this basic side is all these are carbons. It has no fatty acids. Okay, so that's, even though it's a lipid, but it's a lipid because it has lots of carbon. That's what makes it. Some of the steroids, the biggest one that we contact is cholesterol, and cholesterol is involved with making our sex hormones, steroid hormones, and so forth. So we're going to talk about cholesterol, the male and sex hormones a little bit, and steroid hormones, and we won't talk about these two, but these two are steroids as well. Okay, let's look at cholesterol. So this is the chemical structure of cholesterol. Okay, so take a look at the structure in the name a little bit. Okay, so if you notice, it has lots of carbons in it, and it only has 1 oxygen, and that oxygen is a hydroxyl, and you notice it has OL, which tells you it's a alcohol. So cholesterol technically is alcohol, but it has a lot of carbons in it. And the ratio of carbons oxygen is way greater than 3 to 1, so this is very hydrophobic. Okay. All right, so where's cholesterol? Cholesterol is found all over the place, okay, and it's used in all kinds of things. So sometimes cholesterol gets a bad rap. When you go get your tested for cholesterol, and you say you shouldn't have much cholesterol. You need a certain amount of cholesterol because it's found in your cell membranes. It helps make vitamin D, make your steroids, and so forth. Okay. Right. Steroid hormones. Okay, there are 2 types: sex hormones, adrenocorticoid hormones. And what I'm going to do is I'm going to show you the basic structure. And then what we're going to do is we're going to talk about their basic, about their function a little bit. Next. Okay, so here are 1, 2, 3 hormones that are involved with sexual reproduction. And we'll talk about this guy in a second. Okay, the first one is testosterone. So let's take a look at it. A little bit about, you won't have to memorize these structures. Basically, just kind of have to know their function. And a little bit about their names and how they're related. So testosterone. Okay, it used to be called androgen. Okay, andro means man, gen means produce. So what we found out, 100 years or so, is that men tended to have a lot of this structure, and so they thought it's what made a man a man. Okay, so andro, man, gen means make. So they thought that this was the thing that made a man a man. All right, after we've done a little bit more about its chemical structure, we found that it's produced in the testes, so we call it test. It's a steroid, and it's a ketone. Right here. Okay. So the name tells you a lot. All right. Okay, over here, number 2 is structure of estrogen, also known as estradiol. Estro means to bear babies. Okay, or to have babies, and gen means to produce. So produce babies. That's what, and they found this in women. Estro means have babies. Gen means to begin. So estradiol gives me a little bit more about its chemical structure. So it's estra, which means baking baby, and it has 2 hydroxyls on it. So it's di, which means 2 alcohols. Okay, here's structure of progesterone. Okay, progesterone, pro means before gest, so before having baby. It's a steroid, and it's a ketone right here. It's produced in the ovaries to help line the uterus, and also to tell the body that you're pregnant. Okay. Now, this guy here, norethindrone, is a synthetic version of progesterone. Okay. Now, this will be found in birth control pills, and when you take this, your body will basically think that this is progesterone, and when your body when it makes enough progesterone, it's going to stop the ability to become pregnant. Okay, because when you become pregnant, what you want to do is expend energy of just making baby. Okay, that one baby or 2 babies or whatever. You don't want to get pregnant while you're pregnant. And that's what happens that's what progesterone, that's one of the jobs it does. So when you take this guy, it basically tells your body that it thinks it's pregnant even though it's not. Okay, so those are the sex hormones. The other type are adrenocorticoid steroids, and there's 2 types, aldosterone and cortisone. And let's look at them. Again, you won't have to know their structures, but let's look at them anyways, and their function. Okay, cortisone or cortisol is produced, and so on your kidneys, on your kidneys, when you go to your bladder and so forth. Okay, on top of your kidneys are adrenal glands, and adrenal glands produce these 2 guys, cortisone and aldosterone. Okay, when your body's under stress, it's going to produce cortisone, and what that's going to do is it's going to increase the amount of blood glucose in the blood, which means you'll have more energy available to you, and it's also going to increase the glycogen levels in your liver. Okay, and typically cortisone is made in like flight or fight reaction. So if you're getting chased or being threatened, you're going to be producing a lot of cortisol. And it has both bad, if you have too much of it, can really mess you up. If you don't, or if you don't have enough, it'll mess you up. The other one is aldosterone, and this one, it will, it helps increase the amount of sodium in your and water in your kidneys. Okay, if this is too low, you will have low. Your sodiums won't be. Your sodium levels will go low, and then you can cramp up, and also helps you hold water better. I don't know, and so it's used to help regulate the sodium in water in your kidneys. Now, a synthetic version of both of these is prednisone, and prednisone will act like cortisol, because when cortisol is released in your body, it will actually make your blood vessels dilate better, and because it's that fight or flight reaction. And that's kind of what prednisone does. It reduces inflammation, and can treat asthma. What it does is it sort of opens up your blood vessels, so that you can get more oxygen and breathe better. But it also helps with healing, which is related to all of this. Okay, and maybe you'll get into that A&P. Okay, so that's it for the steroids, and we got one more section. Well, actually two. The first one is how do lipids move around in the blood? Now, if you think about it, blood is mostly water. Okay, and lipids means they're polar, and then lipids are nonpolar, and that's like putting oil in water. So if you put oil into your blood, what's going to happen? It's going to kind of maybe clog up your blood, because it doesn't dissolve in water and can't move around. But lipids are nonpolar like oil, and they have to be transported. So how do they move around in blood, which is mostly water? Okay. If you just drop the lipid in the water, it would just lipid in the blood, it would clog up. Okay, it just ball up, and then you die, because the blood doesn't move. So there has to be a way that the lipid is transported in your blood without it clogging up. Okay, and the way it does that is to use something called lipoproteins. And lipoproteins are going to kind of behave like soap, which you're going to see in a second. Okay, so this is here's a picture of a blood vessel that's been clogged up with lipids, and here's one that hasn't. Okay, so for some reason, this blood vessel didn't do a very good job of hiding the lipids. Well, this one did a real good job of transporting the lipids without the blood noticing it. Okay, so we're going to look at this. We're going to look at proteins. There are a couple proteins here. We're going to talk about proteins in the next section, test number 4. Okay, so lipid proteins. Okay, and there's going to be 4 types. The first type will combine as one, and it's called the chylomicron and very low density lipoproteins, VLDLs. Okay, and their job is to move triglycerides, cholesterol, and phospholipids to the cells for storage or energy. Okay, so they move it for energy, all right, and storage. So they move it like the adipose tissue. Okay, LDLs, low density lipid proteins, they have a very specific job. What they do is they take cholesterol to the tissues, maybe to drop the cholesterol to make cell membranes, to make steroids, sex hormones, or vitamin D. So that primary job is to carry cholesterol to tissues. Okay, and a fourth type of lipoprotein is called high-density lipoprotein. And what it does is it's like a scavenger, kind of a garbage truck. It goes around, and so if for some reason some lipids have been dropped, are floating around inside the blood vessels, maybe causing and if there's too much lipids lying around, what they can do is start coating the vessels and start blocking the vessels. And so what HDLs do is they go around and collect excess cholesterol and triglycerides and take them away and take them to the liver and out the gallbladder and the bile salts. Okay, so let's take a look at a little bit how. So the way that they work. So you kind of think of it as like the Pac-Man type thing. All right. So a lipoprotein is a giant molecule, okay? And the outside of this giant molecule is polar. And so when water sees the outside of this guy, it just sees, it likes it. It likes the outside. But inside it is nonpolar, okay? And so what happens is these lipoproteins will, so if you have a piece of cholesterol, so when a lipoprotein sees a cholesterol, what it'll do is it'll grab a hold of the cholesterol and it'll put it inside this nonpolar mouth. And essentially, it will hide the cholesterol from the outside world, okay? Remember how soap works? That's the general idea. And then what it does, it takes it to the cell. And then when it takes it to the cell, okay, that cholesterol, whatever, will be released and sent inside the cell. All right, so that's how lipoproteins work. Is it's that they're kind of like a Trojan horse where they can hide things in it, and make the rest of the world think that it's okay, and then they can move it around. So these lipids can move around without having any damage in your blood vessels. Okay, the final thing is talk about a diagnostics. Okay, so if you get a lipid profile or panel, what they will look at is they will look at your cholesterol and triglycerides. Okay, and you will be given, usually when you're given your cholesterol, you'll be given 3 readings. You'll be given your total cholesterol, your LDLs, and your HDLs. All right, now let's go over these and let's be clear about what they mean in terms of diagnostic and how they relate and how the wording might be not totally correct. Okay, if you've ever had your lipid panel done, you will have heard that your LDLs are called bad cholesterol. Okay, that is really a bad name because LDLs are neither bad or cholesterol. They're a protein that carries cholesterol. Okay, it turns out if you have high levels LDLs, it says that you may have high levels of cholesterol in your blood. So it's just a way of determining if you have high cholesterol in your blood. Okay, now, so that's it. So remember, LDLs are just a diagnostic, okay, but they're actually good because they do have a job. But when you read them as a diagnostic, high levels tell you maybe not so good in terms of the amount of cholesterol. Remember, LDLs are not cholesterol. They're a way of measuring if there's excess cholesterol. So there are ways of reducing your LDLs. One is diet. So if you have a high fat diet, you will tend to have high LDLs, especially our Western diet, and also your genetics. Okay, which you really can't change. All right, I've had friends that have really high cholesterol LDLs, and they have a really strict diet, and they still can't bring down their LDLs all that much. Here's what I talked about, right, and so forth. Okay, now HDLs are often considered good cholesterol. And again, that's actually a bad name because it's neither good. Well, LDLs are good and HDLs good because they're just found in your body. But what this tells you is if you have high HDL, that means you got a pretty good way of getting rid of cholesterol. All right. So it's HDLs are not good cholesterol. All right. Just say you have a good system of getting rid of cholesterol that's free floating in your blood. Okay, and you want to have a high level of HDLs. An exercise and diet can increase those. So when you do have blood work, these are kind of standards that we go by. That they change yearly or annually or every 10 years or so. But they say that your total cholesterol, which is sort of a combination of your LDLs and HDL, not completely, but almost, should doesn't mean there's a law, okay? They like it to be. If it's above 200, it suggests that you may have a increased event of a cardiovascular event, okay? 200 to 220. If you have your LDL, normally should be below 130 for the same reason, and your HDL should be higher than 40 for the same reason. Okay, so the LDLs tell you the cholesterol, that kind of tells you that the cholesterol that's floating around in your blood, and the HDL tells you how good it's taking that away. All right, and okay, and that ends the PowerPoint for this for lipids.